Good afternoon, everyone. My name is Madeline King, and I'm a PhD student in Dr. Lapanaite's lab. It is a distinct honor to introduce Dr. Viviana Gradinaru, a neurotechnologist whose work is transforming how we study and treat the brain. She serves as the Lois and Victor Trundle Professor of Neuroscience and Biological Engineering at Caltech, where she also directs the Richard N. Merkin Institute for Translational Research and the Center for Molecular and Cellular Neuroscience of the Chen Institute. Recently, she was selected as a Howard Hughes Medical Institute investigator, one of the highest distinctions in biomedical research. With a background in physics, Dr. Gradinaru brings an interdisciplinary lens to neuroscience. Over the past 15 years, her lab has developed some of the most widely used tools in modern neurobiology, including cell-friendly optogenetic inhibitors, developed whole-body tissue-clearing methods that render organs transparent, and engineered AAVs that cross the blood-brain barrier after a single intravenous injection. These vectors are now used in hundreds of laboratories worldwide and are advancing gene therapy strategies for disorders that once seemed untreatable, ranging from Parkinson's to sleep disorders. Dr. Gradinaru is also an enthusiastic mentor and community leader. As faculty director of Caltech's Clover Center, she freely shares protocols, viral vectors, and hands-on workshops. She has published over 100 publications with over 30,000 citations, underscoring her field-wide impact. Her accomplishments have been recognized with prestigious awards, such as the NIH Director's Innovator and Pioneer Awards, the Presidential Career Award for Scientists and Engineers, and most recently, the 2024 Carnegie Great Immigrants Honor. Please join me in welcoming Dr. Viviana Gradinaru.
Thank you, Madeline. Appreciate it, and I feel I need to justify my position here. I feel like I'm at a concert and I'm the introductory act before the grand finale. So before the Taylor Swift concert, we will talk about obviously the most important organ in the human body, because all of us, or most of us scientists, we think that what we ourselves study is the hardest, the most interesting and worthwhile subject to work on. And of course, for me, that would be the brain, although the eye is a very, very, very close second, okay? Like, no doubt about that. However, the brain underlies all of our behavior, and it's responsible for everything from how we approach climate change, how we deal with wars or prevent wars, our approach to empathy and pandemics, and even fate as personally described or experienced, as I was reminded during a rather special and magical trip to the Vatican. So all of this is underlined by an organ that's rather fragile. And there's a large disease toll, 1 billion, but this is an underestimate because what I have there doesn't contain substance abuse that, especially after the COVID pandemic, skyrocketed, which puts the toll to almost 2 billion worldwide. So this is why with my group at Caltech and collaborators over maybe a decade now, we focused on a rather, well, we thought foolishly simple topic, but 10 years after, I'll tell you how little we did and how much is left. We focused on how to understand access to the brain. So today I hope to tell you why the blood-brain barrier is important. And we've already had a great morning session talking about the BBB with Carol and ataxia. So I will provide my puzzle pieces to this very, very big challenging puzzle on how to get access to the brain, to understand natural behaviors, and maybe to improve the outcome of brain disorder. And I feel that I should maybe, and this is for the trainees in the room, maybe tell you how I fell into this area, because much like Carol, high-powered chemist, and then taking all of these wonderful tools and knowledge to the brain, I used to be, and I think I still am, a neurotechnologist, an engineer of sorts. And in my past training, I focused on methods to perturb and understand the brain. And some of the graduate school work was back at Stanford with Dr. Dizeroth, where I spent quite a few years on the very modest problem of trying to make microbial opsins not kill neurons. Halorhodopsin, in its first form, would aggregate and kill neurons. So did ARCH, so did MAC. So when you look at EMPHR or EMAC or EMPHR 3.0, those have zip codes that help these bacterial microbial opsins navigate around the cell, make it to the plasma membrane so you can inhibit them at will. And I don't know how many of you have seen that circling mouse with the blue light looking a bit confused. This was like maybe my 200th surgery in the Dysart lab, but it was a very rewarding one, very fun projects to work on. And that foray into optogenetics, so that took us into the importance of maps, just as we heard from Hong Kui, how important it is to know the cell types and their composition. It is extremely important to understand how they actually connect. And that took me to the second neurotechnology part that I contributed in terms of tissue clearing. And those experiments, both in optogenetics and tissue clearing, were so fun because you had so much power, and you've seen in the talks today, so much power to intervene in genetic models, create genetic models of disease, rodents, and image circuits, modify circuits, influence animal model disease outcome. So the realization was that we are so powerful when it comes to mouse genetics, and we are so impaired when it comes to primate genetics and human disease access and understanding. And this is why, with the group and collaborators, we focused on this one problem. And we realized that to understand natural disease behavior, we do need to have distributed access and comprehensive access to these circuits in the brain and spinal cord, and especially for delivery, starting with delivery of genetic tools. And this seemed to be a rather difficult problem. However, we're fortunate because it turns out that each one of our brain cells is within 20 micrometers of a vascular supply. And in total, we benefit from 400 miles of vascular highway, or 650 kilometers for the European trained here like myself. And this makes it a great venue to deliver research tools and therapeutics, except the adult brain is a rather difficult target because, for the better, not worse, it's protected by the blood-brain barrier, a physical barrier comprised of endothelial cells forming tight junctions. And these tight junctions are reinforced by astrocytic enphytes and pericytes. And it's not a barrier. I like to call it more of a portal because it does allow selective crossing, allows nutrients, allows immune defenses. However, it blocks pathogens. So most viruses and bacteria, of course, there's always exceptions, unfortunately, cannot gain entry. So, only allowed agents are permitted. However, there is a way to cross the blood-brain barrier because there are specialized blood-brain barrier receptors, which can enable selective permeability for blood circulating agents. So things that get in our gut or circulation, they can make it to the brain. And this was actually the possibility that was recruited to package DNA in vectors such as the adeno-associated viral vector that led to a wonderful therapy. For example, for children with spinal muscular atrophy, Emily here was treated with the gene therapy for spinal muscular atrophy that uses the AAV9 vector. And because of this, Emily can go ahead and do things children are supposed to do, like playing soccer and playing with their friends, rather than being bedridden and having difficulty eating. The catch, though, is that AAV9 works very well for young mammals. For young Emily, works well. For the more mature blood-brain barrier, doesn't cross as well, and it's also mainly astrocytic. So then we asked ourselves in the lab, sorry, I have to move the cursor again. Oh, the monitor used to have the arrows, so then I could just point this. Oh, okay, actually, never mind, don't touch anything. Don't touch it. It's like my lab folks, oh my goodness. One day they kicked me out of the lab. I was like, Viviana, don't touch anything, break it. And I wasn't mad because we did the same. In the Dysart lab, we had some journalists coming and was interviewing Carl, and Carl took the journalist to the laser room And Carl wanted to turn on the laser, and then we realized, oh, we need to help him. So, but here's the problem. Here's the problem in accessing the mature blood-brain barrier. We have very limited knowledge for the BBB transporters. Of course, transferrin has been studied for this role. It's notable to mention that as a molecule has been known for decades, more than 4 decades, and as a BBB shuttle has been refined and investigated for about a decade. And fortunately, starting with last year and this year, there's very good progress around transferrin, and it's very promising, and it's encouraging for all those being too close to brain disorders. I do think one shuttle might not solve all diseases and problems, and we should expand the understanding around the BBB and diverse shuttles. For obvious reasons, transferrin is present in all of the organs. It's an iron transporter, binds iron, could lead to anemia because of that. So its endogenous role and the natural biodistribution are creating challenges in its best deployment. So then what do? We set up to try 2 things: to cross the blood-brain barrier and to understand how the blood-brain barrier works. And this is where my training as a maybe engineer was very helpful. Before studying biology at Caltech, I was a physics major in Romania, and I fell into optogenetics because of knowledge of lasers and data science and things like that. So then when I was tackling this problem, I realized I can't approach it as a neuroscientist, because the way I was taught in neuroscience was to look at the existing literature and come up with the smartest hypothesis you can and deliver on the hypothesis. But I realized without enough transporters, without enough knowledge, we cannot engineer against those not known transporters. Fortunately, Caltech is a small and unusual school, and out of necessity, you bump into chemists and engineers, and then they think about your problem a bit differently. So we ended up using directed evolution, first in collaboration with my colleague Frances Arnold for optogenetics. So we're using directed evolution. Of course, the audience here is familiar. Frances, first American woman to get the Nobel Prize in Chemistry for using directed evolution for enzyme development. So we used her methods. We learned from her and used her methods for optogenetics first, and then for AAV, to try to pass the blood-brain barrier in rodents and then non-human primates. Okay, so the approach that we took was to use directed evolution, and then evolving these vectors across the species. And then we use these vectors for some basic biology discovery, but the most interesting work that I want to tell you about is to use them to understand how these mechanism-agnostically engineered vectors were actually crossing the BBB. So basically to take the engineered tools to let us know how biology works, to give us fundamental knowledge in the blood-brain barrier. And this work is very much in progress. Then, we are actively working on targeting these receptors for both now rational engineering, so rather than directed evolution, rational engineering of viral and non-viral delivery methods. And we are recruiting some of these pathways within our lab, and mainly with collaborators, though, to try to understand more about brain disease and maybe bring our contribution towards translation. I do have to emphasize, although I'll focus exclusively on blood-brain barrier and the brain, this exact pipeline can be applied to any barrier. The eye, the retina, blood, retina barrier, am I getting this right? Okay. Placental, skin, you can apply it to any barrier and try to find methods to deliver DNA and to understand how those might work. So how did we do it? We took the vector that was used in Emily, AAV9, and we changed basically the the surface of the AAV with molecular methods and created a lot of diversity, right? And the theoretical diversity can be a billion plus. And then we injected these libraries into the bloodstream, and this work was led by former lab member, Dr. Ben Deverman, that now for a while has had his lab at the Broad Institute, driving this work forward, of course. So then we applied selection pressure, I'll tell you a bit about the molecular knobs after, to look at vectors that could cross the blood-brain barrier, okay? So this was the result. So you have AAV9 there. This is adult mouse brain, and you can see AAV9 doesn't cross too efficiently, but then with directed evolution, we could create PHPB that crossed better as good engineers, and well, actually forced by our collaborators telling us we need too much virus, you have to make this better. So then we modified it even more and we came up with PHPEB. During those experiments, serendipitously, we also found some vectors that would better infect the peripheral nervous system. So now for both research and maybe preclinical purposes, it's very important to recognize that one doesn't want full brain delivery of a specific DNA. So because we only modify the viral vector shell, you can intersect this with all of the molecular tricks and power tools that have been developed by the community, including many transgenics from the Allen Institute, enhancers, gene regulatory elements, right, microRNA target sites. So then you can get very good coverage of areas that might be surgically difficult. They might be under the aqueduct, like the dorsal raphe, relevant in psychiatric illness. They might be in the cerebellum, also difficult surgery, or they might span a very large area, such as the GABAergics in the prefrontal cortex in red there. Yeah, red shows up, good. So that's interesting because in that case, it's a DLX enhancer. And to test enhancers, one might need either to make transgenics or do spot injections across the brain, and that's not very efficient. But with these vectors, you package one or a library, although there's some challenges with packaging libraries, and you get the full brain distribution. So now we took these tools in our lab and with collaborators, and we tried to do some basic science. But I think the neurotechnologist in me always sees maybe the technical limitations before the curiosity for the basic science advances. So we realized that actually the tools need to be better for many reasons, but I'll tell you a few. AAV9, when delivered, you see very strong expression in the liver. And so with our tools, with our engineered tools. So what we did here, we worked with Kathy Ferrara's group at Stanford to decorate the engineered AAVs with PET labels and see the biodistribution in living animals. And this was a problem because liver is associated with toxicity. Then we went in deeper. PET is very powerful for live tracking, but it doesn't necessarily give you the molecular resolution and the cell typing. So then for this, we used, we fell back a bit on the tissue clearing that I worked as I was transitioning from my training lab, the Dicerot lab, to my own. So when I was starting my lab again, this is for the trainees, okay? By accident, we made a mouse transparent without the bones and the skin. That got solved later by, for example, Ali Erturk and others. Why by accident? The first version of Clarity was using electrophoresis. Current heats up things and destroys GFP fluorochromes. So we thought, oh, can we do brain clearing without electrophoresis? Electrophoresis would like speed up the micelle flow. So then we thought, why don't we use the same as AAVs? Let's use the vasculature to perfuse Clarity reagents. They were too sticky in their first iteration, so we had to modify the Clarity reagents. We perfused them through the vasculature. And then at that time, my very first technician when I started the lab at Caltech, and then later PhD student with my colleague David Anderson, and then postdoctoral fellow at Johns Hopkins. So I remember coming to my office and said, I think I forgot the transcardial perfusion a bit too long, and we have a transparent mouse. And it was one of those moments like, this is so cool and so useless, but it wasn't like that useless, because it turns out that when we were delivering the AAV libraries, it's one thing to look at the slice in the brain, and it's another thing to look at the entire body. And if you are using the whole body circulation, you want to know everything about that body. Do you get to the brain? Are you, like, do you have residual expression in other areas? It turned out that yes, we had a lot. Yes, we got to the brain, but the liver was still too transduced for comfort, the heart as well. So if you do, let's say, behavioral neuroscience and you look at anxiety, if you affect the heart, you might misread that you're doing something to the brain. So then we went back to our drawing board and said, we need to ask harder questions. Not only we need to get to the brain, but we want to stay away from the liver, from the dorsal root ganglia, from organs that could cause problems. So then, and this is published work, but we went to the vectors and trying to understand their structure and their interactions. Then we did the selections in parallel and multiple Cree animals. And so what we do, the modification on the surface of the vector, we package it in the AAV. And it's under the control of Cree recombinase. So when you deliver to an animal, you only read with your RT-PCR at the end, you only read the vector that crossed the blood-brain barrier, got into a Cree-positive cell, got into the nucleus. We deliver single-stranded DNA. Cree cuts double-stranded, so it needs to get double-stranded, cut and flipped, and only then you read it. So because of all, like in one injection, there's so many checkpoints already. And if you do this in parallel Cree animals, you end up with parallel data sets, and you can look at them and say, yes, we want a vector that's strong in the brain and weak in the liver and DRG, for example. And it turns out that with a lot of loss and mess and effort, it does work because you only need a few good vectors and you start with millions of them. So this is summarizing 10 years of work. My students are not happy when I say that. We started the AV9. We modify, when I say an insertion, it's actually can be up to 60 because the AV has 60 viral proteins, and depending where you change, could be up to 60. So AV9, weak in the brain, very strong in the liver, adult mouse. EB, the intermediate generation that I talked so far, strong in the brain, still very strong in the liver. CAB10, strong in the brain, nothing, or almost nothing in the liver, and the same is true of the other organs we looked at. So now, interestingly, then we went back to these data sets and we said, oh, what else do we have? Because there's thousands and thousands of interesting sequences. Do we have cell type bias? Do we have brain area bias? So then we started to look at different cell types, different brain regions, and we learned some very interesting things. We learned, so these are it's a list of sequences. The key point here, these sequences are not that distinct from each other. But then you take them and you package a ubiquitous promoter with a fluorescent protein, and you get very distinct morphology. You get apparently only neurons, mainly astrocytes, mainly endothelial cells. And this is only by the virtue of the surface of the capsid, right? Why might this be helpful? Because AV, it's a mighty virus, but it's also tiny. You cannot package too much genetic material. So then if you want to get cell type specificity, you might need too bulky promoters or too many gene regulatory elements. So if you move some of that tropism on the surface of the capsid, that's very helpful. And also you could dose lower if you don't have sinks in organs that you don't need or cell types that you don't need. Plus, one example is if you want to target, and we're talking about epilepsy, right, in the morning session, if you want to target neurons, you don't want maybe your product to go in potentially antigen displaying cells in the brain that could cause side effects. So that there's those advantages. So over the years, with, again, and I'll mention our NHP collaborators in the NHP work that's following, but we took this vectors from mice. We improved them a bit for rats, although they're not as good as they could be. It's a matter of focus. And then marmosets and macaques and human cells, right? And intersected with gene regulatory elements, transgenics, et cetera, you can get brain-wide access with cell type specificity. So we put these tools out there. We gave them to everyone that asked it. They're on Agene. And I was very happy to see how creative the community got with them. And these are all papers that we had really no role in it besides putting things on Agene. And there's the many ways in which these vectors can be used. Enhancer discovery is one of them. Another one is to enable comprehensive brain-wide in vivo genetic screens. So rather than spot injection or one at a time, you can package a library and maybe identify disease-inducing pathways or disease-rescuing pathways. So now in our own lab, because of this work, we've started to attract the interest of MD PhD students. So I was very fortunate to have Acacia join my lab. And again, for trainees, my first discussion with Acacia, this was many years back, I asked her what she wants to do. And she said, I want to be pediatric neurologist. And the problem is that there's no good solutions. So during my PhD, I want to work on a therapy that after my medical training, she's in the program at USC, after my medical training at USC, I want to be able to prescribe it to the children. And I was like sitting there, I took a deep breath, and I knew better than telling her, that's not going to happen. And I said, let's see how far we get. So years after, Acacia got quite far. Now, she finished her PhD, and now she's back at her USC medical training. Before embarking on her dream, she realized she didn't have all the vectors that she needed, because the disorders she was interested weren't affecting only the central nervous system. It turns out that there's like more than the central nervous system that we need to worry about with NS disorders. She also needed better peripheral nervous system variants. So she worked with the engineers in the lab using the same exact methods and modified AAV9 for vectors that get better into the nodos or DRG, so for vagus nerve access. And she needed that because she focused on Friedreich's ataxia, right? So she focused on Friedreich's ataxia, loss of function, monogenic disorder. The frataxin gene is small enough to fit within the AAV. So then she needed access to both the brain and other tissues that are affected, such as the cardiac tissue. So then she chose CAB10 for the brain, and she chose this PNS2 vector with good transduction of the PNS tissues, and she used that in an inducible model of FA, a DOX inducible model coming from UCLA, from Dan Geschwing's lab. And she took the mice and ran the predictable behavioral panel with and without genetic replacement. And I'll show you one example. I'm glad I'm showing this because you already introduced to the beam walking assay in the potassium channel talk. So thank you for that. So the top is a wild type healthy mouse, just crossing the beam. The ataxic animal in the middle, slow, many foot slips and fall. The fall is it's a cage fall, and on soft bedding. So, and then the gene replacement mouse. So you can see there's rescue, there's partial rescue. Partial has to do with two things. When in the progression of the disease this was deployed, so Acacia chose to deliver after the symptoms were apparent. And the other thing is that in this case was the CNS only that was rescued. However, of course, Acacia did more animals and assays. But what's important about this proof of concept experiment, because of these different vectors, one for CNS and one for PNS, Acacia could go ahead and test the hypothesis. Do you need to rescue brain, peripheral nervous system, for cardiac, or both? So without doing the transgenics, she could have these cohorts. And of course, maybe predictably, but also unfortunately, you do need both, right? It's not that surprising. You get better rescue if you have both. Students like Acacia also remind us that we've cured plenty of mice. And the reason why this is important is that there's some evolutionary distance when we go from mice to non-human primates and humans. And then I'll briefly summarize what we are able to do in NHPs. Starting with this was actually a collaboration with the Allen Institute, where the Allen Institute has this very powerful system for slices, for acute slices from either macaques or even humans. And you can test these vectors in primate tissue. So what I'm showing here is a vector that's modified, again, with a very important note. There's no blood-brain barrier involved here. It's a slice, right? But you can see that the infectivity is much higher for these engineered viruses. Now, what about the blood-brain barrier? So we worked with Nancy Ip and also with Corey Miller at UCSD and Nancy Ip at Hong Kong to tackle crossing the BBB in the marmoset brain. And you can see here a very broad distribution in the marmoset brain. And it's a newer macaque. There's still significant difference from humans. So we are also, of course, interested to see how it performs in old world primates such as macaque. So this was a wonderful and long-term collaboration with Drew Fox at UC Davis at the California Primate Center. And we were able to increase the BBB crossing in neonate macaques. So these are still young macaques. The blood-brain barrier is a bit more permissive. It's rather challenging in an academic lab to make the volumes of systemic virus required for very large NHPs. So we did what we could. The images, maybe they're hard to follow brain-wide, but here are a few videos that show that with an IV injection, so no brain surgery, with an IV injection, you get gene delivery across the NHP brain. So where are we now with this work? The tools are deployed for basic research. There's some interesting results coming. By using the tools, we are starting to learn something very interesting about the BBB. Again, maybe not surprising, of course, the researchers that have worked hard on BBB, they already knew that, but it was kind of like shaking to see it back at us so clearly. How much BBB varies across species and strains. And here's one experiment that we did where we took PHPB, the first generation engineered systemic vector, and we delivered it to different strains of mice. And you see in C57, super good penetrance, then some intermediate, but then in bulk C, there's nothing, right? So now these are highly inbred laboratory mice strains, and us in the room, by virtue of our biological variables, we're bound to be, I think, more diverse than these laboratory mouse strains. And this speaks to the importance of understanding the mechanism, so these genetic tools, shall they evolve, and when they evolve, are deployed in the right way. So this took actually our research program on the focus of, can we understand more about the biology? Having engineered these tools, can we understand more about the biology, especially how biological variables affect crossing the blood-brain barrier? And this seems like a big problem. Is not only between brains, but also within a brain, because different brain areas will have slightly different permeability properties. At first, it seemed like a very daunting task, but then we realized actually, maybe not, because we've made some good vectors, industry has made some good vectors, academic collaborators and productive, friendly, wonderful competitors made some very good tools. So now as a field, we benefit from hundreds and hundreds of BBB crossing AAVs with slightly different tropism and properties in different strains of mice and different behaviors in NHPs. So all we have to do, funny enough, is to just ask them, how do you AAVs cross the blood-brain barrier? They don't speak, of course, so we do biochemical matching assays where you have cell lines, you display the receptors, the putative receptors or receptor libraries, you flash the AAVs and look for matchmaking. So, yeah. How am I on time? Okay, I can do this, I can do this. Do I get a discount for the technicalities? So here is something surprising. I like this example because when you cross the blood-brain barrier, you think maybe channels, maybe transporters, maybe. It turned out that the favorite way for one of the engineers viruses to go was an enzyme. We would have never looked at an enzyme, especially carbonic anhydrase. But we looked at the enrichment on the endothelial cells, and we just put it through the spool. And the hit that came back was carbonic anhydrase. This is the abundance of carbonic anhydrase on the mammalian blood-brain barrier. And then you see the control. Fortunately, because carbonic anhydrase family was studied for its pH balancing and association with the bicarbonate transporter for a while, there were knockout mice. So we see that in the knockout mice, you don't get AAV crossing. Why is this exciting? I mentioned transferrin. This is carbonic anhydrase 4, and then CD98, another powerful shuttle that's been evolving a little after transferrin. Look at the liver. I might need to use this now. Oh gosh, okay. So look at the liver. transferrin and CD98 very strong in the liver, but not carbonic anhydrase, right? So now we can take this and expand it, and we increase the throughput of this method, and we are applying these biochemical and SPR methods and biochemistry to basically start to look at rules of crossing to different organs and cell types. And the reason why we can do this is that the community overall has developed mechanism agnostic AVs that get to those places, and we just need to find out how they do that. So now that they do that, we can use that information for two things. One is to engineer the AAVs to bind the receptor we want at the site we want, maybe the conserved site between rodent or human, or maybe just for the human site that might not be conserved between the two. So that's one thing that we're doing, and this is highly enabled by structural work. And yes, AlphaFold, it's a hypothesis generating, it's helpful, it's not ground truth. But this method allowed us to now switch from blind-directed evolution to more mechanistic engineering of these AAVs, where we can try to toggle liver detargeting with accessing what we want. So we've done, I'll just skip this in the interest of time, but this is important. AAVs are wonderful. There's also big challenges, big immunogenicity challenges, packaging challenges. Now that we know more receptors for BBB, and we know the peptide engagement rules, because these engineered AAVs, they're peptides that we insert or substitute, they bind these receptors, these new receptors. So we can take that peptide knowledge and now put it maybe on antibodies, maybe on ASOs. So I'll show you proof of concept that you can conjugate IgG to the carbonic anhydrase with a chemical linker, and then you can get across the brain in a very good way. In this case, we actually use atezolizumab, which is an approved drug for non-small cell lung cancer. This cancer is in the periphery, but it metastasizes to the brain. It works to clear the cancer in the periphery, but it doesn't get to the brain. So this creates this possibility. Can we take maybe the existing therapies in the periphery, and by modifying them, one, you cross the blood-brain barrier, two, you get into the cells of interest, maybe with a bispecific antibody, can you start to access the biology of interest? So that's where we are. The final reason why understanding BBB might be important has nothing to do with my work, but has a lot to do with the pandemic we've been through. Viruses and bacteria evolve. The brain is protected. However, for example, bacteria evolved to use transferrin receptor to get to the brain. And we're fortunate the SARS-CoV-2 didn't evolve to lock yet into blood-brain barrier receptors. But I think it would be good to know all of the weak spots of the BBB. Those weak spots could be therapy delivering, but could also be pathogen accepting. So it would be good to know them. So then we anticipate, generate chemical blockers, maybe peptide blockers, to get ahead of potential threats. That's all I had to say. So understanding BBB would be helpful to deliver brain therapeutics of various types, to navigate health threats from viruses and bacteria. I want to emphasize how important it is to take into account biological variation. Yes, it matters, disease state, sex, age. Also, we're more alike than different. That also matters, so we kind of need to always balance it and not overdo it. So this is all I mentioned, the building blocks of the work. Reminder, the same approach can be used for other body barriers, eye, placenta, etc. This is my group of super hardworking scientists, engineers, neuroscientists, everything in between, working hard to basically deliver. So I think that's all I had, and I hope there's some time for questions.
Hi, thank you for your talk. It's very amazing. I'm Jingyi, so I'm the postdoc at Dr. Golkowski's lab in here. So I do have a question. It's amazing to see that across the different species of the mice, they have different resistance to the viral deliveries. Do you also see some kind of differences in the different brain areas at this moment? The second question is like, Because like I know like during the night, the cerebral spinal fluid, they also like flush the brain and wash out like the wastes. And I don't know if this, I would like to know your insights on using the viral vectors delivery to break the blood cerebral spinal fluid barrier and deliver like the potential therapeutics drugs on that.
Both wonderful questions. Thank you. The first question, whether we see heterogeneity in delivery across brain areas, for sure. Some of it we think is due to differences in the blood-brain barrier. As a first, some areas do not have a blood-brain barrier just to allow sampling, right, of circulatory, like the subfornical organs. That's one. Some areas have more vascularization, so if you do have more vascularization, you might deliver more virus. Plus, there's also the aspect you cross the blood-brain barrier, but then you need to get into the cell types. So I spoke simplistically about these vectors. All of them use multiple receptors for different purposes. So depending how they interact, you will see a very diverse behavior. And all of the engineered vectors, like the ones that cross the blood-brain barrier, there's nuances to them. A challenge early on, and I remember when my colleague David Anderson wanted to use it, we weren't hitting the hypothalamus very well, right? So there are some areas that are still difficult. So that's the first question. And the second question, the CSF, we actually try to look at the ependymal cells. Am I getting this right? For the ventricles, and to see whether we could target them to affect the biology that you're mentioning. Now we have something that's positive. Like when we were working, and now we have. It's just like I should probably, if you're interested in it, and if you want to try, we actually have it. We haven't done anything with it because. NIH, thank you, NIH, though. Thank you. But still, so that's one. And then CSF is very important because when we are trying to do delivery and when the vectors were not efficient, right, early on, we looked at all the tricks. And I remember reading this paper where the scientists kept the rat upside down, delivering. So we tried all of these things that have to do with clearance, like the time of the day, right, to just minimize the flashing of the brain. So all of these variables matter, but I think they do start to be in the noise if you can lock into a very efficient receptor. And I think that's the path we want to go towards because of robustness. Rather, this is like diminutive additions that we need, like variables that have an impact, but it's not that strong, because then you get too much variability from one experimental subject to another.
Hi, Dr. Gradinaru. I think fantastic work. So impressive what you've been doing. I wanted to ask about this last portion of the talk where you talk about more of these dissecting these new novel capsids into peptides that could further be harnessed to penetrate the blood-brain barrier or other barriers. Do you think that these peptides could be harnessed to infect these different cell types outside of a harboring motif, so outside of like an antibody coupling, just as cell-penetrating peptides, or perhaps coupled to already existing nanoparticles? I'm asking this question because we're working on delivery for the eye, and perhaps we can try to make like a hybrid molecule, but I'm not sure how these peptides exactly could work.
It's a great question, and actually our collaborators for PET imaging wanted to use these peptides to deliver PET labels across the blood-brain barrier. And it's interesting, most of them didn't work, and we thought is because in the AAV you have the repeats and you benefit from avidity. You benefit from avidity, but then if you just take one peptide with the marker. But most recently, it turns out, so for example, a peptide that it's the one that I showed the NHP slice. That vector binds LRP6. It's a very strongly distributed receptor and a single. So when we move that peptide from AAV9 to AAV2 and maybe 5, it carried, the property carried. So it's basically these motifs can be very specific in their context, or they can be more robust. So the answer really depends on which one, and we've started to prioritize motifs that are a bit more robust across serotypes and across modalities, including antibodies. Or, as you mentioned, LNPs, right? Can you redirect some of this, maybe bigger packaging, safer delivery modalities with this knowledge? If you know the peptide, you know the receptor, can you now go beyond the AAV and mitigate some of the AAV risks that are there for sure? So, yeah, it is an approach. I think it's worthwhile trying. I hope you do it.
Please join me to thank our